Welcome, Dr. Thompson.
Thank you very much for having me along. You can see the slides okay. So I'm a neurologist working in Oxford, and I both look after people with motor neurone disease, also known as amyotrophic lateral sclerosis, and I also do research into the disease. And as you no doubt will all know, measurement really is key to making empirical science work. And what I do, the type of research I do, involves trying to measure this disease in people living with it as a means to really primarily to accelerate our ability to develop treatments. So that's mainly what I'm going to focus on today. But I will talk a bit about some of the other work that we're doing that really, but it all centers on measuring really. So first of all, what is ALS, or amyotrophic lateral sclerosis? Many of you will have some familiarity with the disease. Mostly in the UK, we refer to it as motor neurone disease. And so we tend to use the two terms interchangeably. Amyotrophic lateral sclerosis is probably a more precise description of the disease pathology. So when we're talking about the disease scientifically, we tend to use that term more often. And what motor neurone disease is, it's a disease of the voluntary motor system. And we think of that system as being made up of two sets of nerve cells and muscles. So we have what are called upper motor neurons. These are nerve cells that live within the brain, in the cortex of the brain, and they send a long projection down into the spinal cord or into the bottom parts of the brain. In the spinal cord, or the lower parts of the brain, in the brainstem, they connect with another population of nerve cells called lower motor neurons, which in turn send a long projection out from where they live in the spinal cord or brainstem. They send a long projection out to the muscle. And these muscles, so these lower motor neurons run all the way up and down the spinal cord, sending projections out to the different muscles of the body, including, everything from the muscles of eye movement, which actually strangely aren't really affected by motor neurone disease, to the muscles of the face, muscles of the tongue, muscles that do swallowing in the throat, muscles of the limbs and the trunk, the muscles that control breathing and movement really. So and the way that we, in very crude terms, the way that we think about the generation of voluntary movement is you have activity within these neurons in the motor cortex, the upper motor neurons. They send electrical signals down the upper motor neuron to the lower motor neuron. It passes across a small gap to the lower motor neuron, and then down the lower motor neuron to muscle, where the muscle receives a signal telling it to shorten. And that's how voluntary movement is generated. Now, in motor neurone disease, really, we think of it in a fairly restricted way as affecting these two cell types. It's probably, both how we conceive movement happens and the disease is probably a bit of an oversimplification, but in the clinic, it works pretty well. So motor neurone disease is quite restricted to affecting the upper motor neurons and the lower motor neurons. So the way that the disease manifests primarily is with progressive weakness. It's what we refer to as a neurodegenerative disease. So like Alzheimer's disease and Parkinson's disease being two very common neurodegenerative diseases, it's caused by loss of those nerve cells. So in motor neurone disease, loss of the upper motor neurons and lower motor neurons, they wear out, they die prematurely. So motor neurone disease, as I mentioned, manifests mainly as progressive weakness, and that can affect the muscles of speech and swallowing. It can affect the muscles of the upper limbs, causing weakness of the arms. It can affect the muscles of the lower limbs, causing weakness of the legs, difficulty walking, etc. And it tends to begin in one part of the body and gradually spread over time to neighboring areas. Eventually, it affects the muscles of breathing. And it is really, that is the kind of function that we can't get around in the way that we look after people with motor neurone disease. We can help if they get difficulty with speech and swallowing. We can help with supporting communication and nutrition. If people develop difficulty using the limbs, we can provide help with mobility and upper limb function. And we can support breathing, but we can't completely take over from breathing. So motor neurone disease is a fatal disease. and that is generally because of its effect on the muscles that control breathing. In some people, motor neuron disease is also associated with, or ALS is also associated with, involvement of some other areas of the brain, most commonly other areas of the frontal lobes. So the motor cortex, where the upper motor neurons live, is in the frontal lobes. The other parts of the frontal lobes are more involved in complex social behavior. And so actually people can develop an overlapping condition called frontotemporal dementia, which really typically affects behavior and sort of cognitive functions around social interaction. It's relatively uncommon, so it affects maybe up to 10% of people with motor neurone disease to a significant degree. But the point is, although we think of it as being restricted to the motor centers of the brain and spinal cord, it does affect other areas of the brain as well in some cases. Now, as I mentioned, motor neurone disease is a fatal disease. And one of the challenges about motor neurone disease, which I'll talk about more, is that it is incredibly variable in how it affects people, both in terms of where the disease starts and the types of symptoms that it causes as a consequence of the weakness, but also in the rate at which the disease progresses. And that is exemplified really by how long people live with motor neurone disease. So this plot here is known as a Kaplan-Meier survival curve, and it's a way of charting how long a group of people with a disease will live, or till some event happens. But in this case, it's how long people with motor neurone disease live from the time that their symptoms are first experienced, so from when they first develop weakness. And what it shows is for every person with motor neurone disease, each time the line moves down, that represents one person dying. And along the bottom, the x-axis, it shows the duration from when their symptoms started in months. And so actually, 50% of the people that we see with motor neurone disease will succumb to the illness within around 3 years of their first symptoms. So this is a very, it is in a lot of cases a very aggressive disease. But actually, what you can see from the fact that this line drops to halfway within about 3 years, but actually it carries on and the slope sort of bottoms out or flattens off. And this just is an indication that although a lot of people will die from motor neurone disease in a relatively short period of time, that won't be the case for everybody. So 50% of people will live longer than 3 years. And in fact, around 20 will live longer than 5 years, and about 10% of people will live longer than 10 years. And this is reflected in some of the famous faces who have been affected by motor neurone disease. So I don't know if anyone wants to chip in with naming the famous face, if we want to make it a bit interactive.
David Niven.
Yeah, so David Niven, he died of motor neurone disease in 1983. He first developed symptoms in 1980. So he had what we would regard as a relatively average rate of progression from first symptoms. A more recent famous face, any offers?
Doddy Weir.
Yeah, that's right. So this is Doddy Weir, the famous Scottish rugby international who died of motor neurone disease in 2022 and set up a foundation which has raised a lot of money, raised a lot of awareness of the disease, and has really pushed forward research in a lot of ways. He developed motor neurone disease in 2016, so he lived for 6 years from the onset of his symptoms. So he's in one of the sort of slower progressing groups. And then finally, probably the most famous British person with motor neurone disease, I think, of recent times prior to Doddy Weir, offers.
Stephen Hawking.
Yeah, so Stephen Hawking lived for over 50 years from when he first developed symptoms. So this gives you an idea of the range of the way that the disease manifests, really. One of the other things that we're understanding increasingly about motor neurone disease is that genetics is important in the disease. And that's true of virtually every disease that you could mention, is we know that genes on some level are important. In motor neurone disease, in ALS, it seems as though this operates probably at several levels. So we know that there are families where motor neurone disease happens in a relatively large proportion of the family. So in that case, we know that the disease is attributable to a single abnormal gene, where every child has a 50% chance of inheriting that gene from their parent, and if they have that gene, they have a relatively high chance of developing motor neuron disease or the related type of dementia, frontotemporal dementia. So when we see people where there's a history in the family, so if someone comes to the clinic and they have ALS or motor neuron disease, and they have a parent who had motor neuron disease, we can identify what that abnormal gene is that caused both of those people to get motor neuron disease in about 60% of cases. But we know that there are a proportion of people who will have that kind of history where there's a very strong family history of the disease, but we don't know what the gene is. We also know if we look for those genes in everybody with motor neuron disease, regardless of family history, we know that we will pick up some of those genes in a small number of cases in people who don't have a history in their family. So telling us that these genes are important, but they're probably not the whole story, and telling us that actually if you have one of these genes, it doesn't sort of guarantee that you will develop the disease, but we know that they're very important. We know from some other studies where you look at twins that actually these having one abnormal gene is not the whole story in terms of the genetics of motor neuron disease. We know that even in people where there's no history in the family, where we don't identify these genes, we know that genes are important as well, operating at what we call a polygenic level. So a mix of having lots of genes, some of which increase your risk, some of which decrease your risk. If you've got the wrong kind of balance of those, we know it increases the risk of motor neuron disease. We also think that there's probably something in between those two things. So it may be that people with motor neuron disease have a handful of genes that together have a bad effect, or if you have none of those things, then you're unlikely to get the disease. But it's been very hard to kind of prove that. And over and above genes, we know that other things are important. So probably there are some environmental factors or modifiable factors like cholesterol and things like that are emerging as potential factors that alter risk of developing motor neuron disease. And we also recognise that chance plays a big part in who gets motor neuron disease as well. So it's not very easy with our current sort of technology or what we know at the moment. It would be very difficult to predict who in the general population is really at risk of motor neuron disease. So these create some challenges for how we study the disease. I mean, and one of the other big issues about motor neuron disease that makes it quite hard to study is that it is considered a rare disease. So the lifetime risk of motor neuron disease is around 1 in 400, which, it doesn't sound like it's that rare, but actually in the context of something like cancer or Alzheimer's disease, which will probably affect about 1 in 3 people in their lifetime, you realise that actually that is quite a rare disease. So next, I thought I'd talk a bit about why it is difficult to find treatments for ALS, for motor neuron disease. At the moment, we have one treatment that is licensed in the UK for disease modifying effect in motor neuron disease. It's called riluzole. It has a very small effect of prolonging survival in people with motor neuron disease by really a small number of months. So, it is doing something, but it's really not doing something at the kind of level that we want to be treating the disease at. There are some sort of signals of hope on the horizon, which I'll talk about a bit later. But at the moment, that's kind of where we are for most people with motor neuron disease. So a lot of the work we do in Oxford is centred on really trying to understand motor neuron disease and why it happens as a way to really move on to developing treatments. A lot of that centres around how we do trials, but I'm going to talk a bit about the background of how it is that drug discovery kind of happens and the barriers to developing treatments for motor neuron disease. So the first point is that the nervous system is complex. So it's the single most complicated structure that we know of in the universe. Every one of us has around 100 billion nerve cells in our brain. And there are probably a similar number of supporting cells as well, known as glia. So that's a similar number of nerve cells in every one of our brains as there are stars in the Milky Way. And over and above that, each of those nerve cells has something in the order of 1,000 connections with other nerve cells, meaning that there is about 100 trillion connections between nerve cells in the nervous system. So it just gives you an idea of the kind of scale of the complexity of the nervous system and trying to understand something. I mean, it really kind of takes us beyond the level of the kind of simple treatments that seem intuitively appealing, like, you know, plumbing in a new set of motor nerves, for example, sounds like a kind of appealing approach to treat the disease. But the complexity of the nervous system is such that with current technology, that is way out of the realms of possibility. Another point about ALS is that we understand it to be a complicated disease. It's not. The mechanisms of ALS affect cells in probably many different ways, and it's quite possible that it is not really one disease. So what we know from the genetics of motor neurone disease is that it may be that motor neurone disease reflects lots of different, you know, can happen because of many different changes in motor nerves that kind of coalesce on the motor nerves wearing out prematurely and motor neurone disease. And this image is really just to illustrate the complexity of that there are thousands of different functions that cells perform to work, and nerve cells perform to make us work as humans and for us to carry out voluntary movement. And there are probably, you know, at least 20 or 30 different of those functions that cells do that have been implicated in causing motor neurone disease. And it's not even just restricted to the motor nerves. There is probably involvement from some of those supporting cells as well. And there are people who work in the field who think that muscle may be important as well, and even other systems of the body that are contributing. So really, it's an extremely complicated problem. There's a lot that we know about motor neurone disease, a lot we've learned from disease models and from studying genetics and from studying populations that we've understood about motor neurone disease. But it is an incredibly complicated disease, and that is also a challenge for how we develop treatments. When we really, at the moment, you know, we see somebody in the clinic with motor neurone disease, I can't tell you which of these cellular problems is happening that's causing the disease in that individual. So this is something that we're sort of trying to understand, but I'm not going to talk so much about that today. So that's the kind of complexity of the disease. Then I wanted to just give a little overview of how drug development happens and what the pathways are to developing treatments. The first step is that we have to understand why the disease happens, identify a target, meaning, you know, what is the pathway in a cell that we think we need to attack to reverse the problem that causes motor neurone disease. Then identify potential drugs using screening techniques, you know, taking thousands of drugs, targeting that pathway and seeing if we can reverse what we think is the problem in motor neurone disease. Then you test those drugs in some models to see if they work. And then you take those models into humans and see if you can show that the drug works in people. And actually, in motor neurone disease, despite the complexity of the disease, over the last 25 or more years, actually, we've identified quite a lot of these targets. There's been a lot of drug screening that's gone on that's identified candidate drugs that have looked promising in disease models to validate. But the real challenge and a lot of the cost of developing treatments and the point where a lot of these candidate drugs fails is really at the clinical trial stage. And I think a lot of the reason for that is because of the challenges of measuring a complex disease in a biological system, that biological system being a human being. And that's really what I'm going to talk about. And I think probably it's the bit that's kind of most relevant to your I guess at least the name of your institute, the Institute of Measurement and Control. Perhaps not the control bit, but certainly the measurement bit. This is really what I'm interested in. So I've said this about 10 times already, that ALS is a complex disease. It's complex in how it manifests in an individual in terms of the kind of problems it causes. It's complex in how it manifests in terms of it can involve other parts of the brain. So this is the brain of somebody who had frontotemporal dementia, and you can see this is the frontal lobe here. And you hopefully can appreciate that the sort of bulk of the brain there, these are the gyri of the brain, where the nerve cells live, are sort of smaller and shriveled compared to the sort of back parts of the brain, indicating that this part of the brain is sort of selectively shrunk in someone with frontotemporal dementia. It's also complex in the way that the disease manifests. Another one of the challenges, or a big motivation for working in this area and trying to improve the way that we measure the disease, is that drug development is incredibly expensive. So it costs in the range of billions of pounds to go through that sort of cascade of drug development, and a lot of that cost comes in the clinical trial phase. So as soon as a drug company or an institution takes a drug into humans, the costs rocket enormously. So this is the average cost in millions of US dollars for a drug trial at the different phases. So phase 1 here is really safety studies in a small number of people, a handful of people. Phase 2 trials are where we try and look in a small number of people for evidence that a drug might work. And a phase 3 trial is where you definitively show that the drug does something useful. And that's really where regulators base their decisions about licensing drugs. And the costs are huge. So there was a large trial in ALS a few years ago of a drug called dexpramipexole. It cost around $100 million to run that trial. So you can see that the costs of doing it are eye-watering. So it's imperative really that we know, or we're really confident that a drug is going to be effective when it reaches those later stages, because what we're aiming for is a drug that's effective. We really want to know that drugs are effective, and we need people to be able to continue this sort of pipeline. And if funders and pharmaceutical companies are putting large sums of money into drugs that don't work, then it's going to be harder to find drugs that do work. At the moment, the way that we measure motor neurone disease for the purposes of clinical trials is really to measure the rate at which disability is accrued using a rating scale called the ALS functional rating scale. And this is a questionnaire with 12 questions, where you get a total of a maximum of 48 points if you're completely well with no weakness in any area of the body, and 0 if you were unable to move anything at all and were reliant on a ventilator for breathing. So it's a fairly crude scale, but it has become the kind of standard way of measuring function in motor neurone disease, and it is the standard outcome measure for clinical trials. This graph here shows individual trajectories of around 5,000 people with motor neurone disease who were enrolled in clinical trials over around a 15-year period. And on the left-hand side, it shows the score in an individual. Each line shows the score as it falls as the disease progresses, and on the x-axis it shows the time from when their symptoms first started. And going along with what I've shown you about survival, you can see that the disease progresses at different rates in different people. So there are people here in whom over many months this disability score has changed only a small amount, whereas there are people in this part of the image where their disease has gone very rapidly from somebody with a high score to somebody with a very low score. And that variability is a big problem for clinical trials, because it's very hard to kind of statistically show an effect of a drug where that kind of variability exists, because the groups of treated and untreated people in a placebo-controlled trial are going to overlap enormously. The other things about this scale that make it slightly uncomfortable are that it being a scale that where we're effectively asking people questions about their function, it is quite dependent on both the person sort of receiving the answers and the person giving the answers, so on the patient, on how they are feeling. So it is susceptible to the kind of vicissitudes of being a human living with a chronic disease. So it will depend a bit on how that person felt when they woke up that morning, how well they slept the night before, what's been said to them in the clinic on that day, what kind of a journey they had to get there. So it's a difficult scale to use. It is contributed to by a lot of variability, biological variability, that's uncomfortable. Now, another way that people have thought of measuring motor neuron disease, which is intuitive, is to measure muscle strength, because that is the kind of really the cardinal symptom of motor neuron disease is progressive muscle weakness. So people have developed multiple different ways of measuring the disease. This technique that is on show here in this photo is something called handheld dynamometry. So the examiner there is holding what is effectively a force measurement device, and they are asking the research participant there to bend their wrist back at maximum strength, and they're trying to overcome that strength. But these measurements are also very difficult to work with for all kinds of reasons. So like the disability rating scale, I mean, probably even more so, it's susceptible to day-to-day variability in how somebody's feeling, how well they've slept, etc., if they are unwell for reasons that may be completely unrelated to the disease that we're trying to measure. It also depends quite a lot on the rater. So when measuring muscle strength, if a muscle is of normal strength, the examiner may not be able to overcome that muscle strength. So you wind up with a kind of maximum value of that muscle strength that is dependent on the person who's doing the examining, not on the person who's being examined. So it gives you statistically very difficult to handle data and very noisy data. So I'm just going to give you from that same set of clinical trial data here. One of my research fellows has plotted measurement of one particular muscle, one that's not that difficult to study. It's a muscle in the hand, but measuring that over time in around 1,000 people with motor neuron disease in a clinical trial. And what you can see again is that, yes, you can see that the strength of that muscle decreases over time, but the data is very noisy. In some people, the strength appears to go up before it goes down. In some people, it starts low and goes higher and then comes down. It's extremely messy data. It's very difficult to use, and it's quite impractical. And I mean, and that's kind of over and above the fact that these things are difficult to administer in an objective way, and patients really hate doing this. Probably the most kind of reliable anchoring and one of the most important things that we measure in motor neuron disease is survival. It's really one of the key, if not the key, outcome measure of interest. But as I've already said, it's very variable. And so enrolling people in a clinical trial, it's very hard to predict how long they will live. And actually knowing that and being able to predict when somebody might die of motor neuron disease, or what proportion of people are going to die during the course of a clinical trial, is very important in terms of being able to demonstrate whether a treatment's having an effect or not. And the consequence of that and all of the variability of the measures that I've talked about is that trials in motor neuron disease have to be very long or have to involve very large numbers of people. And both of these things are difficult in a disease that is disabling and life-shortening, because clinical trials involve people taking part in research, coming to the hospital more frequently than they would, taking a drug that potentially causes them side effects, all during a very difficult period in their life. And so none of these outcome measures really is ideal. And I think we recognize that in order to show that drugs work effectively, we do need to know that they have an effect on slowing down the rate at which disability accrues and making people with motor neurone disease live longer, that is a key thing. But at the moment, we're in a situation where we have lots of candidate drugs, but we don't know which of those is the drug that should be going first, be going into a big trial. So this brings me on to talk about biomarkers, and this is the field of research that I work in. And biomarkers, I mean, you may already be familiar with biomarkers, but I'm just going to give a little overview of what biomarkers are. So, you know, biomarker is a portmanteau of biological and the words biological and marker. So really, it's ways of measuring a process, yeah, and in our case, a disease process. And generally, we can do that using either types of scanning technique like MRI scans to look at the structure of the nervous system and the way the system connects together, or physiological testing that measures the electrical activity of the brain, or we can use measurement of the chemicals within bodily fluids. And that's the main focus of the kind of work that I do and what I'm going to talk more about. And when I talk about bodily fluids, I mean primarily blood or cerebrospinal fluid. So cerebrospinal fluid is the fluid that bathes the brain and the spinal cord. So really it is the closest that we can get to the nerve cells that are affected by motor neurone disease. So it gives us probably the most faithful reflection of what's going on in those cells. But it is a bit more difficult to access than blood. Blood, you know, everyone's had blood testing done. Spinal fluid is accessed through a lumbar puncture. It's not a dangerous procedure, but it is a bit more involved than having a blood test, and it's more difficult to orchestrate on a routine basis. But it probably does get us closer to the information that we're really keen on capturing. Part of the reason that we're really interested in biomarkers is because they are objective. So they're not, I mean, to some extent, they may be influenced by some of the factors that I've mentioned, but they are much less susceptible to that, to being influenced by how somebody feels on a given day, for example. In the ideal setting, well, biomarkers generally, we would only recognize something as a biomarker if it is related to the disease process. But ideally, we want it to tell us about the mechanism of the disease and what's going on. It's a way of getting us closer to the disease process than asking somebody a question about how they're affected by their symptoms or measuring their muscle strength. We want our biomarkers to respond to changes in the activity of the disease, but not be susceptible to changes from other things. And we really ideally want a biomarker to tell us, to be linked to those key outcome measures that we're really looking to understand, which is what the disease is doing to somebody, how long that they're going to live for, or how aggressive the disease is, how much weakness they're going to get, etc. And we also, you know, a lot of this is about thinking about how we identify effective drugs and how we provide evidence that they are effective. And so one point is that we need the evidence supporting our use of biomarkers to be robust and robust enough that regulators are convinced that it's telling us what we think it is telling us. Now, the front-running biomarker in ALS at the moment is a set of proteins that we can measure in the blood and the spinal fluid called neurofilament proteins. And at the start, I mentioned that the upper motor neurons and lower motor neurons have long projections that connect them and connect them to the muscles. And these long projections are called axons, and these axons are a big component of the structure that allows them to kind of function and not kind of disintegrate our so-called neurofilament proteins. And there are probably 4 of them. And actually, in motor neuron disease, these neurofilament proteins are released from motor neurons as they die into the spinal fluid, and they make their way into blood, and we can measure them. And there's one in particular called neurofilament light chain, this one here, NFL, so called because it is shorter than its siblings, that we can measure very reliably now in blood and spinal fluid. And as I say, it's released at very high levels in motor neuron disease. Because it's released by damaged nerve cells, it's not specific to motor neuron disease. So in other diseases of the nervous system, these neurofilament proteins go up. So other things where you get damage to nerve cells, for example, Alzheimer's disease, we see elevation of these proteins. Multiple sclerosis, where you have inflammation damaging nerve cells, we see elevation of these proteins, but not to the degree that we see them elevated in motor neuron disease. And that is probably because of the rapidity of the disease and the fact that it almost exclusively affects these very long nerve cells, which have a lot of neurofilaments in them. We know that in people with motor neuron disease, neurofilaments give us a window on how aggressive the disease is. So this plot is a bit similar to the one I showed you previously about how long people with motor neuron disease live. But in this case, people have been divided into 3 groups according to whether they have high levels of neurofilament proteins in red, intermediate levels of neurofilament protein in their blood, that's yellow, or low levels of neurofilament light chain protein in their blood, they're the green group. So here, where the line is falling rapidly in the group with high neurofilament levels, that reflects a larger number of people with motor neuron disease dying in a shorter period of time and having higher neurofilament levels. So i.e., what I'm saying is that having a high level of neurofilament predicts a shorter survival in motor neuron disease. Having a low level of neurofilament protein predicts a lower or a more prolonged survival in people with motor neuron disease, and actually outperforms all of the other measures that we've looked at. So measuring other chemicals in the blood and spinal fluid, looking at some of the clinical features that we know are associated with a more rapid disease process, neurofilament outperforms all of those things. Neurofilament levels are also quite closely, sorry, this is not the right slide, but we know that if we measure how quickly somebody accrues disability with motor neuron disease, so looking at the slope of that previous graph here, if we look at the slopes of these lines, that tells us how aggressive the disease is. We know that the neurofilament levels are also very closely related to that. And over time, neurofilament levels are pretty stable in an individual. So this graph here shows, again, time on the x-axis and the level of neurofilament light chain protein measured in blood. And each of these individual lines shows the trajectory of neurofilament proteins in an individual person. This more solid line with the grey border shows the trend of neurofilaments overall across the group of people. And what you can see here is that although there is a very wide range of neurofilament levels here in people with motor neuron disease, ranging at the bottom end from something like 200 picograms per milliliter up to the over 1,000 picograms per milliliter, the level in an individual is pretty stable. So it varies very little with the disease as the disease progresses. And once the disease is established, these levels are pretty stable in an individual. And actually, that's a very attractive feature of a biomarker, because intuitively that means if we do something, we know it's closely related to how aggressive the disease is, how active the disease is. If we intervene with a drug that improves the disease process, that slows it down, we would expect this neurofilament protein levels to come down, and to potentially come down quite rapidly. Now, that with one caveat has been difficult to show in motor neuron disease, because we don't have a treatment that is effective at slowing the disease down. But in other diseases where neurofilament levels go up, people have given treatments and been able to show that neurofilament levels come down. So one of these diseases is actually related to HIV, the virus that causes AIDS. And in some people with HIV, the virus infects the brain and causes cognitive symptoms. So there's a disorder called HIV-associated neurocognitive disorder, which really means that HIV is affecting the brain. It causes dementia in people with HIV, in a proportion of people with HIV. It's quite an aggressive disease, like motor neuron disease, like ALS. the levels of neurofilament are very high in those people. And if you treat them with treatments that target the virus, those neurofilament levels very rapidly decrease over the course of about 3 months to almost normal levels. And with that, people's function of their brain improves. In multiple sclerosis, the inflammatory disease, which I've already mentioned, you have slightly elevated levels of neurofilament proteins. If you treat people with treatments that modify multiple sclerosis by damping down the immune response, you see that neurofilament levels here come down again fairly rapidly, whereas people who are not treated, those neurofilament levels remain elevated. So there's evidence from other diseases to suggest that neurofilament, if you treat the underlying problem, that the neurofilament levels come down. And it's important to recognize that these are a measure of the rate of the disease happening, the aggressiveness of the disease, not a measure of the distance traveled. So they don't relate to the amount of disability that people with motor neurone disease have or the amount of weakness. They relate to the rate at which the disease is moving. We've done some modeling work using this previous data that we produced to look at if, instead of measuring the amount of disability that somebody's living with using this symptom questionnaire, if we just measured neurofilament levels in blood and used that as the outcome of a clinical trial, what would that do to the number of people that we needed to recruit to a clinical trial to show that a treatment, an effective treatment, was effective? And really, we sort of modeled this statistically and showed that you really reduce the number of people that you need in a trial pretty enormously, by half. And this is a fairly conservative estimate, so we could really potentially streamline trials. And also, because we think neurofilaments would decrease rapidly based on the other diseases, probably you could shorten trials a lot. So we could both have trials that run using a smaller number of people, but that also run over a much shorter time scale. And that is beneficial for lots of reasons in terms of cost, but also in terms of how tolerable it is to people living with motor neurone disease. Now, I mentioned that because in motor neurone disease we don't have a drug that works, we don't have really evidence to kind of show that what we think is likely to be the case with neurofilaments, we can't show that it is the case. There is a slight exception to that. So in 2022, there was a drug trial published that gave a treatment to a subgroup of people with a rare genetic form of the disease called SOD1 ALS. So this is people who have motor neurone disease caused by abnormalities in a gene called SOD1, and this was the first genetic form of motor neurone disease to be identified in the 1990s. And so, 25 years after the discovery of that gene, this trial was concluded where people were given a treatment that sort of targets that gene and stops the abnormal protein from being produced from that gene. It's now SOD1 ALS is a little bit different from what we refer to as sporadic ALS, in that when you look at the nerve cells affected by the disease under the microscope, they look a bit different. But in the clinic, it's the same disease. And interestingly, in this drug trial, they found that it was very difficult to show a change in that functional rating scale that we talked about in the disability scale. It was very difficult to show an effect at 6 months, which is how long the trial really ran for. They only began to see effects on the disability score later. But the striking thing for me and for a lot of people, including the regulators in the States and in Europe, was that they had a very rapid effect on the levels of neurofilament light chain protein measured in blood. So within 3 months, the levels came down to nearly 50% of what they were at the start of the trial in people who were given the drug, but didn't decrease in people who were not given the drug in the placebo group. And by 6 months, it really had gone a little below 50%. At the 6 month time point in the trial, they gave everybody who was on placebo also got given the drug, and you can see similar to the people in the who were given the drug initially. Those people in the open label extension who only got given drug at 6 months onwards, their levels dropped as well. So for me, this is really a fairly compelling evidence that using neurofilaments would be a great way to study the effect of a drug in a group of people with motor neurone disease. So along with my colleagues in Oxford, Professor Kevin Talbot and Professor Martin Turner, and so Martin Turner really has driven a lot of the research into neurofilament proteins over the years, starting more than a decade ago. He had certainly been thinking about this approach for a number of years, and based on the work that we've done modeling neurofilament proteins and the work that Professor Talbot had done in Oxford, working with a group in Edinburgh, identifying a candidate drug called terazosin, which is a drug that's already licensed for the treatment of prostate symptoms and blood pressure. We have set up a clinical trial in Oxford that is based purely on measuring the levels of neurofilament light chain protein at the start of treatment, so before people get the drug, and then at 3 months and 6 months after they are treated. The outcome that we're looking at being, we're hoping to see if the drug is effective, that it lowers the levels of neurofilament protein in the blood. So this is the first time that anybody has done a trial in this way, so it's a fairly pioneering approach, we think, and it's actually now led on to a larger multi-centre study that's happening across the UK that's called Experts ALS, where we're kind of expanding this approach, and we're going to be using neurofilament levels as a way to measure multiple drugs being tested simultaneously in a group of people living with ALS. And this is a big study that's being funded by the National Institute of Health Research, and is, I don't know if you are aware of the £50 million that was promised by the government last year to fund motor neurone disease research, but actually a chunk of the money from that is being contributed to this research study, Experts ALS, which has come about really as a direct result of this work using neurofilament proteins as a way to measure the disease. So neurofilaments really are the kind of are at the moment are the kind of big thing in terms of how we're measuring motor neurone disease and what we think is the way forward in terms of clinical trials at the moment. But as I've already hinted, there's a lot about motor neurone disease that we don't know, and there's a lot that we want to understand about how a problem, a specific problem in cells of the nervous system in one person might cause motor neurone disease, and a different problem in cells of the nervous system might cause motor neurone disease in a different person. So the idea that there are kind of multiple tributaries that can get into the final common pathway of motor neuron death and motor neurone disease in the clinic. So one of the reasons that drugs may have failed in motor neurone disease, and this is something that neurofilaments may not be able to overcome, is that it's possible that the drugs that we're using are targeting pathways that might be relevant in some people, but they're not relevant in everyone with the disease. The idea that maybe it's we're trying to treat what is effectively multiple diseases with one drug. So a lot of the work that I do centers around analyzing cerebrospinal fluid to try and understand in an individual what is going on, what are the drivers of the disease in that person, so that we might be able to better match the right drug or drugs to that person to kind of treat their disease. So, and I use a technique called proteomics, or mass spectrometry is what I center around mostly. And some of you here, I don't know exactly your backgrounds, but some of you may have experience of using mass spectrometry in the engineering realm. But mass spectrometry we use as a means to measure lots of different constituents of bodily fluids. It's used across the kind of biological sciences, but in my case, it's really used primarily to measure cerebrospinal fluid proteins. And we use a type of mass spectrometry called liquid chromatography tandem mass spectrometry. And effectively what we do is we take our bodily fluid, we take cerebrospinal fluid, we treat it to I'm interested mainly in looking at the proteins, because proteins are kind of the effectors of cell functions. They're effectively the machinery of cells, is the proteins, and they give us a window on what is going on in cells in a person, and they're relatively easy to measure because they're quite stable. But one of the difficulties is that fluids like cerebrospinal fluid are very complex. There are thousands of proteins in there, and actually individual proteins are quite difficult to measure because they're chemically badly behaved. And so one of the ways that we overcome this, and so maybe it's a bit difficult to explain what I mean by chemically badly behaved, but for example, some proteins don't dissolve well in water. Yeah, so some proteins will only dissolve in lipids, in more oily substances, and actually it then becomes very difficult to study those two proteins together. And in fact, it gets more complicated than that, because some proteins, because of how complicated they are and the way that they function, in some proteins, one bit of it will dissolve in water and one bit won't. One bit will only dissolve in the more kind of oily, non-polar things, alcohols, that kind of thing. So that becomes a very difficult problem to solve. So actually what we do is we break the proteins down into smaller chunks where we can where we can then make them so that they dissolve in a more reasonable range of solvents. So we make it so that they can dissolve in a bit of a range of those things in a way that we can handle it. And then we inject that kind of mixture of broken down proteins into a mass spectrometer where we measure the size of each of those chunks of protein with very high accuracy, and we then break those chunks of protein up into smaller chunks, and we measure the size of those smaller chunks, and from that we can work out what protein that is, and we can measure the amount of protein that went in. Yeah, so this is a kind of very complicated technique that we use, using extremely sensitive, very accurate machinery, most of which is manufactured in Germany, to allow us to measure proteins. And as a consequence of this, we can measure with quite a high level of accuracy. We can measure thousands of proteins in a single sample of spinal fluid, from around 50 microliters of spinal fluid. So that is about a 20th of a mil of spinal fluid. We can easily measure about 1,500 proteins from one person. And actually, we've been doing this now for a number of years. So this is something that we published in 2018. And just by measuring in this case, it was about 800 proteins in the spinal fluid from people with motor neuron disease and other conditions, we identified a set of proteins that are elevated in people with motor neuron disease. And these are not proteins that relate to motor neurons at all. These are proteins that relate to supporting cells in the nervous system called microglia and astroglia. And these are cells that have different jobs in the nervous system. They're not sending electrical impulses and making things happen, but they're helping the nerve cells that do all of the things that the nervous system does. They're helping them to live by clearing up the kind of rubbish that's produced, the debris, by supporting them with energy and these kind of things. And actually what we found is that at a relatively coarse level, the levels of these proteins that we identified relate to different features of the disease. So some of them are related to the aggressiveness of the disease, but others are related to how well the brain is functioning in terms of the cognitive functions, the mental functioning of people with motor neuron disease relate to other proteins. And it gives us a window to some extent on what these different populations of supporting cells are doing in terms of motor neuron disease. And this is really, this is at the moment at a fairly coarse level, but it's opening a window on what might be the important mechanisms of disease for different people with motor neuron disease. and it's beginning to help us understand where we might target different drugs in different people. But this is still kind of work that's at a fairly early stage, and at the moment we've got a very big project happening where we're kind of expanding this to look at this in a much larger number of people, to look at a much larger number of proteins with higher accuracy, to really try and take this to the level where we hope within years we might be able to take a group of people and say, well, this is the pathway that is relevant to you, and so you should be taking this drug, and then start to use that as a way to try drugs in people where we think there's a much higher chance that that particular drug is going to work in that particular person. So the kind of final bit of measurement I wanted to talk to you about, or the final bit of the biomarker work that we are engaged in that I wanted to talk about, relates to genetic forms of ALS. So as we've recognized that there are genes that predispose people to motor neurone disease, where, you know, if you have a particular form of a gene, you have a high risk of developing motor neurone disease. We kind of unmasked a population of people who, and these really are the children or siblings of people living with motor neurone disease that's caused by a gene, a single gene abnormality. We've kind of unmasked this population of people who now know that they are themselves living with high risk of developing motor neurone disease or that related type of dementia. And that, you know, a lot of what we've understood about the genes and the fact that these genes are there from the moment that somebody is conceived through gestation, through, you know, their time in their mother's womb to birth, as the nervous system grows and develops, through childhood as the nervous system grows more and is refined. They might have this abnormal gene from conception. They live a completely normal period of adult life for, you know, 20, 30, 40, 50 years, and then suddenly something changes, leading to this progressive disease. And so one of the things that we really don't understand is what's going on before this that maybe primes the nervous system for this problem to happen, or that allows their nervous system to compensate or cope with this abnormal gene that's there from conception. So one of the things that we are really keen to understand is what is going on in this period, both because it might enable us to better predict who is at risk of developing motor neurone disease, and therefore intervene at an earlier time as these genetic treatments become available, but also to help us understand why the disease happens, so that we can target treatments to these mechanisms that are happening early. So, for example, if we found out why it was that someone could live for 50, 60, 70 years with this gene, but without getting motor neurone disease, if we worked out what it was in the nervous system cells that was compensating for that problem, that would be a pretty good bet for a treatment, I would think. So some of the work that we do involves now studying people during this period of normal adult life, when we know that they have a gene that puts them at high risk of developing the disease, but that they are living a normal life with a completely normally functioning nervous system. Neurofilament proteins that we've already talked about, we know from a study looking at this group of people that actually when people are well and are healthy, their neurofilament levels are pretty low and are pretty normal. And that it's only really when they are in the short period of maybe 6 to 12 months before they develop symptoms of motor neurone disease that those neurofilament proteins go up. So at the moment, it looks like we can't use neurofilament proteins as a way to predict when the disease is happening, other than in the very short period before it does. But actually, this has formed the basis of a preventative trial in people with that rare genetic form of the disease called SOD1 motor neurone disease. So there is a preventative trial going on currently of people who have that particular genetic predisposition. We're also interested in looking in the more general population as well. So we and others have done work using large population studies, and actually, interestingly, just using things that are collected through routine general practice appointments, cholesterol levels, for example, we see alterations in cholesterol levels that may be linked to risk of motor neuron disease. But we also see, and this is actually a study from Sweden, but we've shown the same thing in the UK, in the UK Biobank study, showing that levels of cholesterol, particularly LDL cholesterol, bad cholesterol, are high in people many years before they develop motor neuron disease, but actually decrease as they get closer to developing motor neuron disease, and then sort of flatten off or even increase as they get very close to developing the disease. And so this may be a way that we can refine and use to potentially predict when people are really at risk of developing motor neuron disease. So that was all I really was going to talk about today in relation to the kind of work, the research that we're doing to try and measure motor neuron disease, to accelerate treatment, and really to try and make motor neuron disease a preventable disease. This is just acknowledges the contribution from our funders and the support from the university. The top photo here is this is not all of our research team, but this is most of our research team that we have in Oxford, and in the bottom panel is the clinical team from the Motor Neurone Disease Centre in Oxford. If you're interested in learning more about the kind of research we do, this is our research website. That's my personal email address. This is our group research email address if you have any further questions that come up that that you don't want to ask today. So thanks a lot for your time and attention.
Excellent. Thank you very much.